Hi, this is Dr. Mandy, and I am here to talk to you about why I love the BiomeFX stool test. Before I begin, a quick disclaimer. This is a functional analysis of the gut microbiome, and it's only intended for educational purposes and not to be used for diagnostic purposes. Microbiome research is still in its infancy, so we encourage you to think of this report as a piece of the puzzle rather than an answer sheet. And special thanks to Microbiome Labs for creating this wonderful option for patients who want to evaluate and support their microbiome. So why do I love this test so much? There's so many stool tests out there and they're all wonderful, but I happen to gravitate towards this one. And why is that? Well, it's all based on the distinction between the two types of sequencing technologies that exist. One is called 16x ribosomal RNA sequencing, and the other is called whole genome sequencing. The analogy I usually use to explain is that the two is that 16s ribosomal RNA sequencing is what the vast majority of stool tests use, and it's limited because what you're doing is you're looking at, let's say 6, 7, 8, or different variable regions of the bacteria's DNA. And if you find them, if you find 2 or 3 or 4, you might put them together and say, okay, this is likely a bacteria. The analogy is often, for example, if you look at somebody's elbow and their knee and their chin, you might think that that might be the whole picture of somebody, but in reality, you really didn't get the whole analysis of what that person is. Now, whole genome sequencing means in order to identify bacteria, we have to look at the entire DNA sequence, and we have to find that entire DNA sequence before we identify the bacteria. So we've gone from this primitive sequencing technology, which is that 16S, which is now very, very cheap to run. And it's probably the main reason why most of these companies still use it, because you can run it for, let's say, $20, $30 a sample versus the whole genome, which is still more expensive because the technology is newer and it requires a lot more to do. So let's talk about the functions of the gut microbiome. The microbiome performs a variety of functions that I can't explain all of it in this short presentation, but in a nutshell, there's processes such as the production of short-chain fatty acids and hormones and neurotransmitters to anti and pro-inflammatory compounds. And the gut is responsible for digestion and assimilation of essential nutrients, and the microbiome participates in this role. Gut microbes can produce some protease enzymes to help promote this things like protein digestion and fermentation in the gut. Now, many fibers are not digestible by the human GI tract and can thus feed the gut microbiome by design, but maldigestion and malabsorption can allow for carbohydrates and proteins to enter and act as substrates for bacterial fermentation. So what you're going to learn in this presentation is that your diet counts when it comes to your microbiome. So let's dig into the test. First page is a snapshot of the report called the report summary, and it consists of different areas of evaluation of the function and health of the gut. Let's go through each of these. The first one is the gut microbiome index. And this is an overall score for the gut microbiome health. A score above 30 is considered excellent and is calculated by assessing 4 key indicators of microbiome health in your gut. And these key indicators are in the next few slides. The first one is alpha and beta diversity. Alpha diversity is how diverse your microbiome is and how many different species your microbiome has. It's tied to reduced risk of all types of chronic condition, better postbiotic production, a stronger metabolome, which means all the metabolizing and microbiome products benefit a healthy microbiome from having a diverse microbiome. So it's low. Alpha diversity is really important. It's basically telling how rich your species are. Low diversity suggests that your gut microbiome was recently damaged by antibiotics, environmental toxins, stress, diet, and other factors. Beta diversity is the variation of the species when comparing 2 separate ecosystems. This marker takes the composition of your gut and compares it to healthy populations in your environment. Basically, how well adapted is your gut to others in your same area, such as the United States, for our purposes. Next one is the resistance that each bacteria have against each other, called the resistance index. It's measured in antibiotics that are produced by bacteria against other bacteria. So this measure is actually a measure of the health and activity and ecological composition of your microbiome. The more of this you have, the healthier your microbiome composition is because it maintains equilibrium. Next one is your pathogen control index, and it's a relative abundance of pathogens, especially in the context of the rest of the microbiome. It looks at a number of specific pathogens, one that are relatively common and tend to be correlated with disease outcomes or symptomology or dysbiosis in general. Basically, they're sequencing about 16 or 17 or so pathogens and doing a full genome sequencing to look for presence of pathogens. And not only are they looking for the disease, but looking at relative abundance to other microbes in the gut. Other microbes in the gut microbiome. Basically, it's relative abundance, the whole ecological mix. Now, please keep in mind, just because something's out of range, it doesn't necessarily diagnose a disease. It doesn't diagnose a disease. It's just correlating. So if you have a lot of diarrhea and your Clostridium difficile is high, then it could be related, but not always. I've had C. diff for a number of years and I've had no issues with diarrhea. So please don't look at this as diagnostic. They're just clues. Obviously, the higher the pathogen indexes, the more in control your body's basically taking care of it. Bacteria via bacterially produced antibiotics. Very important are your keystone species. They're beneficial bacteria that have disproportionately large effect on both their habitat and status of other microbial communities in the gut, and they really deserve a lot of attention. These are protective bacteria, and they produce molecules that act as food for other bacterial species and control the acidity and inflammatory tone of the gut. In a nutshell, keystone species create an environment of healthy balance within the gut. The abundance of keystone species may be relatively large or small, but without them, your gut microbial community cannot prosper. And here are some very important functions of the gut microbiome, which I will summarize next. But before I do, I want to do a little quick overview of your gut microbiome phyla. The major phyla of the microbiome include Firmicutes, Bacteroidetes, Proteobacteria, and Actinobacteria. In order to understand how they relate to each other, it's important to understand the role and players within each phyla. For example, Firmicutes consist of your Lactobacillus, Bacillus, Clostridium, Enterococcus, and Ruminococcus. Now, Clostridia do represent about 95% of the Firmicutes phyla. Harmful strains of bacteria from Firmicutes can become overgrown due to, here's where your diet plays in, excess saturated fat intake, which can negatively affect microbial diversity. This is another reason I'm not a big fan of the high-fat ketogenic diet. Now, Bacteroidetes are generally considered beneficial for the gut microbiome and consist of, so this is the second one, genera such as Bacteroides, don't confuse that with the phyla, and Prevotella. These are gram-negative bacteria that also ferment fiber and other polysaccharides to produce the short-chain fatty acids propionate and acetate that support intestinal health. And they also are linked to improved sleep efficiency and quality. Unfortunately, these are negatively affected by excessive fat and animal protein, but their abundance can be increased with fiber and carbohydrate intake. The next one is Proteobacteria, which is typically found in low abundance in the gut. And increased numbers of these can be a hallmark sign of imbalance within the gut microbiome. This phyla thrives on saturated fat and sugar and contains many opportunistic pathogens. Western diets are known to increase the abundance of the Proteobacteria phylum. And finally, Actinobacteria are proportionately less abundant as well, and mainly represented by the Bifidobacterium genus. You may have heard of bifido strains in some probiotics, and they are a strong butyrate production, which is a short-chain fatty acid. They are actually the first microbes to colonize the infant gut during vaginal birth, and they are positively correlated with breastfeeding and proper immune system development. Sufficient levels of actinobacteria, especially bifidobacteria, have been associated with healthier digestion and intestines. In addition, certain members of actinobacteria are associated with improved cognition, memory, social skills, and reduction in fear response. Let's go through the ratios. The first one is the Bacteroidetes and Firmicutes and Bacteroidetes ratio. Because these are the two most important phyla present, their ratios are really important. A low ratio indicates a healthier metabolism, while higher ratios are associated with unwanted weight gain. Next ratio is your Proteobacteria-Actinobacteria ratio. Ratios below 1 are associated with healthier metabolism and cell turnover. And increasing actinobacteria may also benefit mood and brain health. You can increase actinobacteria, by the way, by consuming plant-derived carbohydrate starches such as polysaccharides, such as FOS, GOS, XOS, inulin, and arabinoxylan. Next ratio is your Prevotella bacteroides ratio. High ratios are associated with lower BMI and reduced incidence of chronic inflammatory disease. Lower ratios are associated with metabolic imbalances and are positively correlated with high intake of protein and animal fat as typical of the Western diet. Higher abundance of Prevotella are seen in individuals that consume rich carbohydrates and fiber. Bacteroides is increased by sugar and saturated fat, while Prevotella thrives on fiber-rich foods like fruit, vegetables, beans, and whole grains. This donut chart depicts the most abundant bacterial phyla in your gut. The percentile chart compares the relative abundance for each bacterial phylum between your gut and the microbiome of typical healthy populations. So it's kind of nice to see that side-by-side comparison. On the side here, we have percentile values between 25 and 75 that are typical. And you can see the low and high if yours are on the lower side or the higher side. So there's two types of fermentation gut bacteria prefer. We've got saccharolytic, which is carbohydrate fermentation, and then there's proteolytic, which is protein fermentation. First is sacrolytic fermentation. Important keystone species such as Ruminococcus brahmi, Lactobacillus, Bifidobacteria, and many others thrive on carbohydrates, specifically resistant starches and dietary fiber. There's a wide range of fibers that feed a specific group of bacteria, and these include soluble, insoluble, resistant starches, inulin, oligosaccharides, and more. Sacrolytic fermentation is also associated with improved metabolism and enhanced gut acidity, which can deter unfavorable microbes. Super important if you have dysbiosis and have pathogens, you've got to start to promote the sacrolytic fermentation. In fact, higher levels of sacrolytic fermentation are associated with a healthy metabolism and reduced intestinal pH, which support a healthy gut environment. Sacrolytic fermentation levels may be low as a result of high protein, low fiber diets, or insufficient keystone species. Sacrolytic fermentation yields short chain fatty acids at a ratio of approximately 20% butyrate, 20% propionate, and 60% acetate. Butyrate is the most important short chain fatty acid. It improves gut function, scavenges ammonia, regulates immune system, reduces oxidative stress, imbalances blood sugar. Propionate is important too. It has a strong impact on the immune system and can be a partner with butyrate. And acetate is used for cholesterol synthesis, lipogenesis, and can be converted to butyrate by some bacteria, but too much can lead to weight gain. The next few slides just demonstrates the different fibers that are best for sacrolytic fermentation. Now that you understand your keystone species depend on these fibers, you can see how important it is to include these in your diet whenever possible. So you can see there's resistant starches, and then there's inulin and the food sources, and then the primary degraders. So by eating these fibers, you're actually promoting the growth of some of these bacteria. And then here's your FOS, GOS, and XOS, which is also found in some prebiotic supplements as well. All right, let's move on to lactate. This test can identify your lactate production and what percentage is L versus D, what percentage of your producers are L versus D lactate producers. Lactate is an intermediate of carbohydrate metabolism produced from pyruvate during lactic acid fermentation. It plays an important role in modulating your immune system and also inflammation. These species use lactate as a substrate to produce short-chain fatty acids. However, if there is an overabundance of lactate producers, as there is in this person, you can see here, paired with low abundance of lactate utilizers, this will cause a surge of lactate in the gut, which can be toxic and harmful to your tissues. Excessive lactate production is seen in a lot of diseases such as IBD, short bowel syndrome, and intestinal ischemia. You can see in this sample, this person is producing a higher than optimal level of lactate, but also has an overabundance of D-lactate producers. Excessive D-lactate is associated with many symptoms such as bloating, distension, GI symptoms, fatigue, air hunger, balance and gait issues, and memory loss. So you really don't want to have too much of the D-lactate. Next up is proteolytic fermentation, and this is generally thought of as less favorable than sacrolytic. as it yields compounds that are toxic to humans, including amines, phenols, and sulfurous compounds. Low dietary fiber can increase proteolytic fermentation due to insufficient amount of fermentable carbohydrates. Shifts occur towards proteolytic fermentation, and when that happens, this can alter the relative abundance of microbial species in the gut and generate bioactive, damaging metabolic byproducts and products. Higher levels of proteolytic fermentation are associated with gut dysbiosis and inflammation. Proteolytic fermentation levels may be low when the gut microbiome is more diverse and utilizes more sacrolytic fermentation. Histamine can be used in the gut and can travel to distant areas of the body, where it may induce unfavorable symptoms. Patients with an overabundance of histamine-producing bacteria should focus on strengthening their intestinal barrier function, because leaky gut can allow gut-derived histamines to enter circulation and promote dietary intolerances and allergies, or disruption in healthy allergic responses. High levels of gut-derived histamine are associated with high abundance of proteobacteria, Roseburia, Morgani, and Klebsiella pneumonia, and also a decreased abundance of bifidobacterium, which is why some of the histamine probiotics, like there's one called probiotic histamine X, they are mostly bifido species to help degrade the histamine. Here's a snapshot of some of the microbes that produce histamine. Keep in mind, there are some lactobacillus and some bifido species, so be very careful with probiotics that you use when you have a histamine issue, making sure that you're not actually adding more histamine in the gut. Phenols. Pcresol, for example, is a byproduct of tyrosine metabolism by gut bacteria and can be toxic to intestinal cells and impair intestinal barrier function. Pcresol is also toxic to a wide range of gut microbiota, especially gram-negative species. When you pair this with a diet in low fermentable fibers, these foods have the potential to dramatically increase phenols and Pcresol, as gut microbes are forced to use amino acids for energy. So one way to reduce these levels is to increase gut acidity through the intake of resistant starches, galacto-oligosaccharides, fructo-oligosaccharides. Those main three starches can reduce the production of these toxic metabolites. Ammonia is next. We all know ammonia to be very neurotoxic. Ammonia is a byproduct of amino acid fermentation by gut microbes. Ammonia is also produced in the small intestine through the bacterial degradation of glutamine. Healthy liver and kidneys can filter and excrete ammonia through the urine. What is known is that an overgrowth of ammonia-producing bacteria paired with excessive protein intake and liver or kidney dysfunction may lead to a toxic surge of ammonia in the blood. High levels of ammonia are detrimental to the brain and the liver. Bacterial strains that lack urease activity can reduce ammonia concentrations in the gut by crowding out ammonia-producing bacteria. Sulfate-reducing bacteria convert dietary sulfur and taurine to hydrogen sulfide, or hydrogen sulfide, a toxic, we also abbreviate that as H2S, a toxic compound that impairs intestinal detoxification pathways and can cause gas that smells like rotten eggs. So if you have gas that smells like rotten eggs, you might be producing too much of this. Hydrogen sulfide production is associated with high protein, low fiber diets. Sulfate-reducing bacteria compete with methane producers and acetate producers for the same substrate, hydrogen substrate. And as a result, it's important to maintain a delicate balance amongst all three. Methanogens convert acetate, ammonia, hydrogen gas, and trimethylamines to methane gas. Methane can also slow the intestinal transit and affects gut motility, which may then allow increased time for nutrient absorption. And as a result, that can cause weight gain, but also can cause potentially, with the slower motility, can cause SIBO. Methane producers compete with acetate producers for substrate utilization, which may explain why methanogens are indirectly associated with digestive issues. GABA is a neurotransmitter or chemical messenger in the brain that blocks specific signals in the central nervous system to slow down the brain. This provides a protective and calming effect on the brain and body. High-fat diets are associated with reduced GABA levels in the prefrontal cortex by 40%, which can result in various mood imbalances and difficulty sleeping. Glutathione is the most important antioxidant in the human body. It is found in nearly every cell in the body and is the primary agent of detoxification in the liver. Glutathione can also act as a hormone, regulating the release of GABA and dopamine. Glutathione is produced from 3 amino acids, glutamine, cysteine, and glycine, which are obtained from food or supplementation. Deficiency in glutathione may lead to production of free radicals and oxidative damage throughout the body. Recent evidence suggests that the gut microbiome determines the levels of glutathione throughout the body. So if you have low glutathione or a lot of free radicals or oxidative stress, well, it could be coming from deficiency from gut microbes, because it says that they can determine the levels throughout the body. Indole is a byproduct of the microbial degradation of tryptophan that can be utilized in a variety of ways in the gut microbiome. Indole can bind to serotonin receptors in order to regulate behavior, gut motility, food intake, and can support immune and intestinal health by interacting with gut microbes, doing things like scavenging free radicals and increasing the expression of xenobiotic metabolites and enzymes like cytochrome P450. So it's an important compound. Indole functions as a signaling molecule that may be increased during latent infections. You can balance indole production, but you should have balanced levels. As you can see, the green is in the middle. So having lower highs is not good. But having too low is not good, as I just explained, but not having too much can also cause changes in mood or cognition as well. So having too much can affect your brain, and having not enough can affect gut barrier, which then can indirectly affect the brain. The Estrobolome is a network of 60 genera of bacteria that can recycle or deconjugate inactivated estrogens for reabsorption into circulation by producing very powerful enzymes. This recycling process is handled by gut bacteria within the beta glucuronidase or beta glucosidase activity. So there's 2 enzymes. When the estrobolome is too abundant, the body is unable to eliminate estrogen effectively, causing estrogen to build up and leading to estrogen dominance, as is probably happening to this person. On the other hand, if estrogen recycling is too low, this may lead to insufficient levels of estrogen in this in the circulation. So either way. All right, so guts and bacteria synthesize vitamin K and many of the B vitamins, including biotin, cobalamin, folate, nicotinic acid, pantothenic acid, peroxidine, riboflavin, thiamine. Those are the main ones. So the production may be low as a result of low alpha and beta diversity. So having adequate levels of alpha and beta diversity will help dictate the levels of vitamins produced in the gut. Let's go through each of these. Thiamine is a vitamin that plays a critical role in energy metabolism, especially in the brain and nervous system. Thiamine also plays an important role in muscle contraction and nerve conduction. Faecalibacterium species utilize thiamine but don't produce it. So there is also competition for vitamins within the gut microbiome, because gut bacteria will also use it. So you have to have enough of the bacteria that produce it, otherwise it's going to steal it. Vitamin B2 riboflavin is a cofactor needed for energy production and fat metabolism, and also plays an important role in immune cell function, and various bacteria are responsible for producing that. B5 pantothenic acid is essential for energy production and fat metabolism. Bacteroides, Fragilis, Prevotella capri, and Ruminococcus, and Salmonella enterica, and H. pylori can all produce vitamin B5 in the gut. However, there are many species that rely on B5 for growth, but cannot synthesize it. So they're also competing. And these include bifido species, Faecalibacterium, Lactobacillus, and others like Clostridium difficile. So that's also an important thing to keep in mind. Vitamin B6 is an incredibly versatile nutrient that supports immunity, brain function, and protein metabolism. Biotin B7, also known as vitamin H, can also sometimes be called B8, is a water-soluble vitamin necessary for growth, development, and cellular production that can support healthy hair, skin, and nails, and support healthy immune responses. Biotin is synthesized from tryptophan by intestinal bacteria, such as Bacteroides fragilis, Prevotella capri, Ruminococcus lactaris, Clostridium difficile, Bifidobacteria infantis, H. pylori, and Fusobacterium varium. But some species of Prevotella, such as Bifidobacterium, Clostridium, Ruminococcus, and Faecalibacterium, and Lactobacillus may steal biotin from the hosts as they need it for survival. So think about it. If someone has an overgrowth, let's say in one of these bacteria like Clostridium, it could be lowering your biotin levels. And this helps with appropriate supplementation for when you're running this test with other tests, for example, like a Nutri-Eval. All right, B9 folate is essential for healthy blood cells. Gut-derived folate is directly absorbed in the colon, contributing to about 30% of the daily recommended intake. If B9 producers are low, there could be low levels of this nutrient available to the body. Cobalamin B12 is crucial for healthy red blood cells, brain, and nervous system function. DNA regulation and metabolism. Production of B12 by the gut bacteria can contribute to about 31% of the daily recommended intake for this nutrient. And finally, vitamin K2 is a fat-soluble vitamin necessary for calcium metabolism and critical for the health of teeth, bones, nerves, and cardiovascular system. Most K2 comes from dietary sources. However, gut bacteria, including E. coli, Bacteroides vulgaris, Bacillus subtilis, and Bacteroides fragilis, can also produce K2 endogenously. However, microbial-derived K2 has a protective role against oxidative tissue and damage in the gut. And finally, I have to mention that your diet does count. And as you've probably learned in this lecture so far, that high protein, high fat diets can be catastrophic to your gut microbiome. However, some of us have certain reasons why we have to follow specific restricted diets. That's why it's very important to work with your practitioner or nutritionist to ensure that you are getting the right amount of fiber and the right amount of prebiotics and other plant-based ingredients to help promote that sacrolytic fermentation to improve your keystone species and other phyla in the gut and those short-chain fatty acids. So if you're following something like a low oxalate diet and you have to watch your intake of various plant-based foods, there are some strategies around that. I have various strategies that I utilize to help people stay within their safe levels while still promoting that really rich bacterial environment for diet environment for the bacteria to flourish so that you have an overall healthy microbiome. And I can see in this picture, we're looking at things like resistant polysaccharides, which are then consumed by primary degraders, and intermediate products are produced, such as short-chain fatty acids. And then we have the butyrate producers, which then produce butyrate. So you see the importance of these resistant starches in the diet. And that's the end of the lecture. I hope that you now have a better understanding of the BiomeFX stool test and why I love to use this as a way to look for clues to come up with strategies to improve your health. Thank you.